Great. Thank you so much, Courtney. So good afternoon, everyone. First of all, thank you for the kind invitation and the introduction. I'm very excited to be here to discuss one of the interesting research directions in the lab, where we have been thinking very deliberately about the underlying properties of the data and the relationships when we try to model it, and more importantly, when we try to integrate across different types of data sets for various prediction tasks in genomics and in the biomedicine domain. So the whole problem or the whole motivation comes from the fact that in the recent years, there has been an information explosion, both in terms of volume and variety. And what is happening is that researchers across universities, clinics, and hospitals are collecting lots of different types of data sets to try and understand the phenomena that they are studying, whether it be a biological phenomena or a disease. And so in this scenario, where earlier in more traditional setting, we would have the understanding of the world and use that to model different types of mathematical models, we have moved a little bit towards data-driven approaches, where the idea is that now we are developing these models using and leveraging the data that we are collecting. And so machine learning plays a very important role here, and our lab basically does machine learning research in developing and applying different types of models. So the basic idea behind machine learning is that we have some individuals and we have some outcome associated with. So because this is biomedical informatics, I'm using this example. So say we are predicting side effects. And so we collect some numerical features, which we represent as our input X. And we also collect information about the outcome that we observe, which becomes our output Y. And so given our observed X and Y, we then try to learn a function fx through machine learning, which connects our input to our output. And the reason we want to learn this function is because if we have a new patient come in and we have the information about their numerical features that we had collected earlier, we can feed it into the function that we have learned. And if we have done a good job of learning this function, we can make an accurate prediction of whether this patient will have the side effect or not. So this particular flavor of machine learning is called supervised learning, and there are many, many models that can allow us to learn this function. But in the recent years, as you may all know, deep neural networks have become very popular in the domain, and their popularity comes from the interesting results that we have seen originally in the image domain, so for the image classification task, and then also in the language domain, where the language modeling was done using neural networks. And the reason is because these models have shown to give very good performance when making accurate predictions. And this is basically, and the way they works is that given this image, we try to learn a function, F1, that connects the image to the output. But now what we do is we stack these functions or the layers one after the other. And so what happens is that each individual layer processes the input and then passes it to the next layer, and that one takes that input, processes it, and passes it to the subsequent layer. This is very roughly based on our understanding of how human brain works. And so the idea is that when you see an image of a dog, the neurons in the first layer kind of processes that information and pass it to another set of neurons that process that information and pass it to another set of neurons. And then eventually you are able to make the deduction that this is the image of a dog. So because of the stacking of functions, what we end up learning is a complicated function that is fully capable of capturing all the complexity that exists in the data set to connect the input to the output, and hence they tend to give accurate predictions. So again, coming back to our task, we have the input, we want to connect it to the output, and now we can use a neural network to learn a complicated function that can do the job for us. Now, the complexity in the input domain space has also been increasing, and this is basically because of the multi-modal nature of today's world, where we try to collect very different types of measurements from the same individual. I'm going to talk about genomics eventually. So we also do this in the genomic setting, where we calculate or we measure different type of genomic phenomena around the DNA. And so now we have this notion where we have multiple different types of data sets, and we want to kind of combine all of these data sets to help us make our predictions more accurate. And so now the machine learning model task not only has to learn a function that connects the input to the output, but it actually, has to do this data integration task of combining all these different modalities to make accurate prediction. There are multiple methods that can do that, and the whole data integration framework falls into two steps. The first step is representation learning. So we want to learn some numerical representation of the different types of data sets that we have so that they are, roughly comparable or they connect well. And one such example that a lot of studies would do is to take a neural network architecture, run the data set through it, and have a numerical representation in the end, which is just vectors or a matrix of real values. And then comes the second step, which is the data fusion step, which is, combining all of these different representations together. And what most studies do is that they will do a simple concatenation or an averaging of the features to combine all these data sets and then move on to the prediction part of the model. So this is great and it works a lot of times, but what a lot of methods have not been considering for a very long time is how do we do more data-aware representation learning? So you can imagine if we fit the same neural network architecture for all the different types of data set, some of them might actually miss what the actual true underlying structure of the data is and may not do a good job of learning its representation. And then the second issue comes at the data fusion step where, you can imagine if you're just doing a simple concatenation or averaging, you are losing a lot of information about the explicit interactions that may exist between the different modalities. For example, this is just an example, maybe the interaction between the modality of gene expression and DNA sequence is more important for a particular prediction than that between clinical information and gene expression. And if we are not explicitly modeling these relationships, the model will kind of miss on this information. So in today's talk, I'm going to talk about two projects where I'm going to show how we do this data-aware modeling in the first task, where we are combining the 3D architecture of the genome and this genomic signal called histone modification to predict the gene expression. And we use a graph convolution network to do that, to accurately model the underlying structure of how the DNA is arranged inside the cell. And then I'm going to shift gear to the biomedical side and talk about this project where we are integrating 3 different data sets to predict the Alzheimer's disease diagnosis using a multi-modal cross-attention framework. So here for the data fusion, we are actually explicitly modeling the interactions between each modality instead of simply concatenating them. And I forgot to mention this in the beginning of my talk, but feel free to jump in and ask a question at any point. It's absolutely fine. So I'll start with the first story. And this project was done by my PhD student, Jeremy, along with 2 undergraduate students in the lab, Zavi and Charlotte. So data integration problem is also important in genomics, and the whole motivation comes from the fact that while the DNA sequence can give us a lot of information about cell development and diseases, it may not be sufficient. Because while all the cells in our body have roughly the same DNA sequence, they're all expressed differently. So the cells in our eyes are very different from the cells in our heart, and so on and so forth. And so researchers are, in order to try and understand the biology, are trying to understand how these different cells are expressed in different states. And a lot of it boils down to the expression of the coding region of the DNA, which is the gene. And depending on which genes are expressed at what time could result in different kind of downstream effects. But understanding whether a gene is expressed or not is a very difficult task because there are lots of factors that play a role in its regulation. For example, you may know about the transcription factor binding, which is a protein binding that turns or signals for the gene to start getting transcribed and convert into proteins. But this is not enough. What could also affect this particular event is where the gene is on the DNA sequence at that time, because the DNA, as you remember, is wrapped around in a 3D space inside our cells. So what happens is that the DNA is wrapped around these proteins called histones. And if these histones are nearby, then that region becomes closed and things cannot come and interact with the gene, and the gene is less likely to be expressed. Whereas if these histones are far away, then things can come and interact with the gene and the gene would be expressed. And this has been related to the phenomena of these histones having tail, and these tails being prone to certain chemical modifications called histone modifications. So there are some histone modifications that result in these histones being far apart, and some modifications that can cause these histones to kind of come closer. And so all of this kind of, plays a role of whether a gene would be expressed or not. And the question we want to answer here is, well, how are these histone modifications related to gene expression? So the reason we would want to answer this question is because, as I mentioned, all the cells start from the same genome, and then over time, the epigenomic environment of the cell changes, and these changes lead to different types of genes being expressed that result in different cell states. And just how these are important for cell development, they can also play a role in, causing disease of the cell. And one thing to remember here is that unlike a DNA sequence, these chromatin or epigenomic changes are potentially reversible. And that is why it's really important to try and study them, because you can imagine we can develop these epigenetic drugs so that we can just target these epigenetic changes, do not touch our DNA at all, we don't have to worry about any mutations being introduced in the DNA at all. So that's why it's very important to try and understand how these histone modifications could be related to the gene expression. And in trying to understand that, we have spent a lot of time collecting data sets. So we have data sets about the gene expression. We also have data sets that capture, whether a histone modification is present and where it's present using ChIP-seq experiments. But the problem here is about the scale of the combinations that we are looking at, because it's not just one gene, it's 20,000 genes. There could be combinations of 20 different histone modifications that could be playing a role. And we are not just looking at one cell lines, we are studying hundreds of cell lines. And so if somebody were to experimentally go and test each and every combination and see its effect on the gene, this task would be impossible. So we decided to tackle this task back in 2016, which is learning the relationship between histone modifications and gene expression using deep neural networks. The reason we decided to do that was because of this property of these layering of the neural networks that allows the neural network to not only capture the local features, for example, when we are classifying this image as a public park, but the later layers of the neural network can also capture the combinations or the complex interactions between these local features to make an accurate prediction. So our idea back at that time was to take the histone modification signals as the input, and we wanted to learn its relationships to the gene expression, so that became our output. And we said, well, the local features could just be the profile of the signal across the genomic regions. And the combinatorial features could be the interactions between these different histone modifications. And because of this property that we wanted to learn, it made sense to use a neural network for this prediction task. To our delight, we showed that we were able to, make accurate predictions of the gene expression from the data, suggesting that we were able to capture the combinatorial relationships between histone modifications. But more importantly, we did not stop there. We actually spent some time doing interpretation on our model, and we found this very interesting result. So what we found was that whenever the model made the prediction of the gene expression to be off or repressed, the features that it considered most relevant were the histone marks H3K27ME3 and H3K9ME3. And this was interesting because these two marks are known in the literature as repressor marks, and so they are known to repress gene expression. And this is not an information that we gave the model, it's something that it picked up on itself. So that was exciting. But one thing we didn't do at that time was we didn't factor in the notion of long-range interactions that exist inside the DNA. And that's not only us, but the subsequent studies that came afterwards also did not do that. So what are these long-range interactions that I'm talking about? Well, I already said that the picture is complex, but let me add more complexity to it. So we tend to view the DNA as a linear structure, but it's actually a structure that is arranged in the 3D space. And so a factor which is very far away from the gene in the linear space could actually be interacting with it just because of its proximity in the 3D space, because the DNA kind of loops up on itself. And while this could be involved in regulation, this kind of interaction could also be involved in misregulation. And this is not just a hypothesis. There are actually evidence of studies over there that have shown that these long-range interactions are involved in different types of diseases. So the reason we didn't involve this information back in 2016 was because these data sets were still coming out, but thankfully now there are many data sets that can capture the 3D organization of the DNA. And one such data set is a Hi-C data. So what this data set captures, it's a matrix where the rows and the columns represent the genomic regions, and the value of a matrix represents how frequently those two regions are interacting. So if it's a high value, that means those two regions are interacting more frequently. And if it's a low value, it means they're not interacting that much. And what we did was we took this Hi-C map and we formulated it as a graph, as it should be, because here in each graph, what we have is the node is representing the genomic region, and we connect two nodes with an edge if there is evidence that those two are interacting. And if not, then we leave it as it is. And we formulated it as a graph, and we said that, well, we still want to use our neural networks, but instead of using some of these existing architectures, we decided to use graph-based convolutional neural networks. Now, you may be familiar with convolutional neural network. What it does is that for a given pixel, we learn a representation of this pixel by taking a weighted aggregation of all the neighboring pixels, and it's very popular in images. Now, graph convolution is roughly the same idea, but here for each node, we learn a representation of the node by taking a weighted aggregation of all its neighboring nodes. So two pieces of information we need to have to make this model work. One is the node features. So these are any numerical properties that represent the node, and an adjacency matrix, which tells us what nodes are connected and what are not. So to give you an example, suppose if you were dealing with a social network where each node is a person, you can imagine that the node features could be the age of the person or whether that person is employed, so some numerical features. And then the adjacency matrix could look like this, where the rows and the columns are the nodes, and we assign a value 0 if they are not connected and a value 1 if the two nodes are connected. Right. So there are many different ways to aggregate the information, and there are very many different formulations of graph convolution neural network. We went with this particular formulation because that was the one that was kind of popular at that time. There are a lot of notations on the screen, so let me walk you through them one by one using the example of this graph, where we are trying to learn the representation of the node that I have marked with a star. So first thing first, without any convolution step, the original embedding of the node is just the original features that we start with for the node. And then we do the first layer of our convolution step. As I mentioned, it's a weighted aggregation over the nodes of the neighboring nodes, and we are normalizing it by the neighborhood size. And then again, this step is specific to this particular formulation, but we also add the information from the previous layer embedding. So previous layer embedding is just the features of this node. And finally, because this is a neural network, there is a nonlinear activation function, a nonlinearity over here. We include that, and that becomes the embedding for the first layer. Now we repeat this for the second layer again, and we do it k number of times, and that gives us the final representation of the node. Now, an important thing to remember here is that the number of convolution steps we do, the further the information away we are capturing from. Because what has happened is at the first layer, the node got the information from its immediate neighbors. but the neighbors themselves got information from their immediate neighbors when we ran convolution over them. So when we repeat the convolution one more time, now we are effectively aggregating information that's 2 hops away. And so if we have k convolution layers, we are getting information that is k hops away from that node. And that's how we aggregate the information. So we call our method GC merge, and we use graph-based neural network to solve the prediction task. So given a particular gene, we want to predict its expression. First, we get the notion of what are the neighboring nodes of that gene using the Hi-C contact map. So that gives us the information of what the neighboring regions are. And then for each node, we get the numerical features as the signals of the histone modification chip-seq for all the histone modifications. So we have the node features, we have the information of the neighbors. This fits very elegantly into the graph convolution neural network framework. And so we run this, we get the representation of that node, we feed it through some fully connected layers, and then we get the final prediction. And so we use the data sets to train the model, and then we make predictions on the test data set. So for the experimental setup, we use 3 different cell lines, GM12878, K562, and HUVEC. As already mentioned, the inputs are the graph structure that we create using the Hi-C map. And the features of the node are these 6 histone modifications that I have listed. And the output that we are predicting is the gene expression. So first thing first, do we make accurate predictions? So we show that the model makes good predictions. The predicted gene expression values are correlated with the true gene expression values. We also compared our method to some existing neural network-based baselines, and we overall saw that the method gave better performance. But this is not where we wanted to start. Just like our previous project, we were again interested in what are the important genomic regions and features that the model is using to make accurate predictions. And so what we did was we incorporated this method called GNNExplainer into our framework. What this method does is, for a graph neural network setting, for a node that it makes predictions on, we are able to learn the importance scores that tells us that what neighboring nodes are most important for that model to make prediction on that node. And we can get these importance scores for features as well. So what features are most important for the prediction of that particular node? And the way that works is by maximizing the mutual information between the model predictions in its original form and the prediction of the model once certain parts of the graph are masked out. So the way that happens is by minimizing this objective function. So what we are doing here is we are optimizing these fractional masks M and N. M is for the nodes, N is for the features. And we are minimizing the difference between the original predictions of the model and the prediction of the model when we mask out certain edges and we mask out certain features. So very intuitively, you can imagine if you mask out or you remove certain nodes and the prediction doesn't change too much, that means that those nodes are probably not important. But if the prediction changes drastically, then you know that those nodes are important. And so you can calculate an importance score kind of proportional to this effect. So we tried this interpretation on a lot of genes in our data set, and here I'm showing an example of 4 such genes. So the yellow circles are representing the genes that we are predicting. The circles around them are representing the top 10 neighboring nodes of that gene, and the size of the circle is representing the importance scores that the model gave it. And the red circles are actually experimentally validated long-range interacting regions for those genes by the study that I have listed below. So what was very encouraging to find is that the importance scores that the model was assigning corresponded well with the truly long-range interacting regions that had been seen experimentally. We also got these importance scores for the features, but there is no way for us to validate these experimentally. But what was interesting to see was the 2 features that the model said were important were H3K27AC and H3K4ME3 marks. And these are respectively promoter and enhancer marks that are known to be involved in long-range interactions. So that was exciting to see as well. Right. So to wrap up the first story, GC merge, it integrates spatial information to successfully predict gene expression. It is able to capture long-range interactions, and the explanations we get can serve as a hypothesis tool by indicating what long-range interactions are relevant and what features are relevant. And the code is available and the datasets are available on a GitHub repo if people are interested. Now, the key takeaway from this story is that like having an architecture that can capture the underlying of the structure, the underlying structure of the data can be very beneficial, and especially in biology where a lot of data sets have these graph-like structures. And so this idea was, very interestingly, was also seen in many other studies. So a study that came out very around the same time as ours from Christina Leslie's lab was doing something very similar, where they had more fine-grained long-range interactions that they were modeling as a graph using a graph-based neural network and looking at fine promoter and enhancer interactions as well. And then, there are many other studies that have since come that do similar type of modeling. Right. So I can take a bit of a pause if there are any clarification questions before I move on to the next story.
A quick question, very interesting story. I was wondering if you had a measurement or assessment of how important relatively the local modifications or the long-term interactions were, like in your graph model. If you just removed all of the long-range interaction data, what would your gene expression prediction look like?
Yeah, that's a very good question. So I think that comes back to the comparison over here. So the CNN and the MLP are basically deep learning and multi-layer perceptron baselines where we did not have the long-range interaction. So here it was basically just looking at the histone modification signals around the region of the gene when making the expression prediction. And so these were the kind of our baselines to say what happens if we don't incorporate long-range interactions. And there's another, because this is all averaged out, so the differences don't look that great, but we also have a result in the paper where we showed that these differences, this improvement was more drastic for highly expressed genes versus the genes that had low expression values. So that's where it made more difference, the incorporating the long-range interactions.
Hey, Ritambhara, I have a similar question. So instead of having the graph, but this time, what if you connect it to the closest enhancer or the closest element instead of, just the distance-based graph? Would you still have lower performance than what you have? Have you tried that?
Yeah, so we have a result which says that I think we did that for the explanations, but not for the prediction. So we tried to show that the explanations that we were getting about the importance, those were not just capturing the distance. So we basically showed the importance score space that the model was producing and compared to the distance of the nodes. And what we found was that it wasn't that the model was picking up only on the closest neighbors, it was actually picking up on some neighbors that were further away than the closest neighbor. So we have those results for the explanations, but not for the predictions.
I see. So when you make your graph, then you don't weight your edges, right? It's based on the
in this study, we did not, but thanks for bringing that up because we have a new paper which is solving a slightly different task, but we have this notion of positional encoding that capture the distance, and we have seen that makes a difference. Yeah.
Thank you.
First. All right. So I will move on to the next study. So completely switching gears here, going more in the biomedical side of things. So as I mentioned in this study, what we are trying to do is predict the Alzheimer's disease diagnosis using 3 different modalities from imaging, genetic, and clinical side. The work was done by my PhD student, Mihal. She's co-advised by me and Karsten Eickhoff. So the idea here is that Alzheimer's disease is one of the most common degenerative diseases. It affects millions of people worldwide. And the tricky part is its diagnosis, because what studies have found is that very few Alzheimer's disease patients get diagnosed accurately based solely on the clinical symptoms. And unfortunately, this disease doesn't have a cure. So all the treatment is more preventative and also focused on delaying the onset of severe symptoms. And so this condition known as mild cognitive impairment, which shows up before the actual Alzheimer's disease diagnosis, is very important to accurately and to diagnose accurately and timely. So given the complexity of the disease, for the past 2 decades, Alzheimer's Disease Neuroimaging Initiative has collected a lot of different types of data sets across different patient populations. So we have the control set, but also patients who have been diagnosed in the MCI category and those who have been diagnosed with Alzheimer's. And because what we have seen is that just clinical data is not sufficient, they have collected imaging data. So for this study, we'll be using the MRI, genetic data, which in this we'll be using the SNPs, and then clinical data sets that have information about the demographic of the patient, but also things like neurological exam scores and so on and so forth. And so the question becomes that given that now we have these different types of data sets, can we combine them in an efficient manner so that we can make accurate predictions about for the Alzheimer's disease diagnosis? And there have been a lot of multimodal studies ever since this data set was released. And they follow the same steps of data integration that I already talked about, which is they learn some sort of a representation of all these different types of data sets. And then they do a simple concatenation of these representations to make the predictions. And as I mentioned, you can imagine that in this multimodal setting, a simple concatenation may miss a crucial component, which is the importance or different set of importance for different cross-model interactions. Maybe it's more important to look at the interactions between the MRI and the clinical data versus the SNP and the clinical data and things like that. So these studies don't explicitly model that. So our approach is to basically follow somewhat the similar setup. So the first thing we do is we have, data specific neural network layers. So for the images, we have a convolutional neural network. And for the other 2 modalities, we have fully connected neural network. And this is not novel. This is something that the previous studies have done as well. What is novel in our study is the way we are combining the data sets. So instead of just concatenating the data sets, we are actually looking at pairwise interactions of all of these modalities. Now, when we do these pairwise interaction, like explicitly model them, you can imagine now the model has to look at a lot of information at the same time. So what we do is we incorporate an attention mechanism to try and reduce the cognitive burden on the model when it's looking at all of these different interactions and trying to make accurate predictions. So what do I mean by attention mechanism? So the idea of attention mechanism was introduced back in 2014. And again, it was roughly inspired by how how attention happens in the human brain. So when you're looking at this image, you can tell that it's a park. But in order to say that, you are not looking at each individual pixel of the image, but actually just focusing on the most relevant parts that give you the most information about what is happening in the image. And so this is what human attention is. And the idea is that can we use some sort of function or mechanism to allow our deep neural network model to also focus on just the most relevant parts of the input instead of looking at everything. So as I mentioned, this attention mechanism was introduced in 2014, and this was first introduced for the language translation task. So I'm going to use that example to discuss what attention mechanism is. So we have, say, some words in English, and we want to, see which word translates best to the French word chien, which means a dog. And so, if you are using a neural network to do this, you will have some numerical representation vectors, Q1, Q2, Q3, and then a vector K for the final output. And again, the simplest step would be to just concatenate all of this information and let the model try and figure out what would be the accurate translation. But you can imagine that, is and walking is not really relevant for the model to make an accurate translation. So instead of doing this, simple concatenation and letting the model figure out, the idea here is that what if we have these extra set of weights, which we call attention scores, which allows the model to give more emphasis on certain parts of the input as opposed to the other parts. And so we calculate these attention scores using this function that I'm showing here. And what is happening here is that this attention, the first step is just taking some sort of a similarity score between, the original words and the word that we are translating. The higher the score means the more important, the more relevance. Lower score means probably not very relevant. And it's a weighted similarity score. So these weights are something we allow the model to learn. And then once we have these similarity scores, we have a softmax function, which converts them into probabilities. So values ranging from 0 to 1, such that everything adds up to 1. So the beauty of this mechanism is that as the model starts seeing lots and lots of examples during training, it will learn these attention weights in such a way that it will give more emphasis to the words that are more relevant for making the predictions and less emphasis to the words that are less important. And so that roughly translates, so this becomes the output now gets weighted by these attention scores. And so the model gives more emphasis to the word dog when trying to make this prediction as opposed to the other words. And so this is, roughly translates to the idea of that the model is giving more attention to a particular entity over all the other entities and reduces the cognitive burden. So coming back to our framework, we call it a MaDDI, Multi-modal Alzheimer's Disease Diagnosis Framework. As I already mentioned, we have these initial, layers to learn the representation. But then once we have learned these representation, we first do our first level of filtering at this step, where we apply what is known as self-attention layer. So here what we are doing is we are doing the exact same mechanism, but for the input with respect to itself. And what that does is that it allows the model to learn the interactions among different parts of an input. And then once we have the output from this layer, for each modality, we model its pairwise interaction with the other two modalities. So for the image, we combine it with the genetic and the clinical, for the genetic with the image and the clinical, and for the clinical with the other two. And because, again, now the model has to look at a lot of interactions at the same time, we apply this cross-model attention layer, which allows the model to learn the most relevant interactions between different modalities. So instead of looking at all possible interactions all the time, the model over time can try and focus on only the most relevant interactions and propagate that information forward. So once we have the output from that, we put it all together, have a fully connected layer at the end, and we produce our output, which is basically classifying whether this patient is from the control group or the MCI group or the Alzheimer's diagnosis. Okay, so we take the data set from ADNI. Because this is a multimodal setup, we actually have a small sample size compared to the individual modality, because the overlap of the patients that have all the three modalities in a decent format is small. The demographics are over here. We wanted to make sure that they weren't extremely skewed in terms of sex or the age of the patients. There are some differences, but we didn't see any extreme skews. We did some pre-processing and feature pruning based on the previous study. So in the end, we have 29 different clinical features, 3 MRIs, and 15,000 SNPs. And we did random training and held out test splits for 90 of the proportion 90% and 10%. And finally, we performed 3-fold cross-validation on our training set to select the hyperparameters. So first thing first, we check the performance of the the CNN model and the fully connected on the individual modality prediction tasks with lots of data sets. And we saw that, we picked our architecture based on the best performing metrics. Now, one thing to note is we just picked our best performing architecture. We never used this data set again when we tried to do multimodal predictions. And so having picked our representation learning architectures, we combined them using our MaDDI framework. And what we showed was that the numbers we got on our test set were much higher than the ones reported before. Now, you may notice that I'm not presenting this as a bar graph, and that's because these are not exactly comparable. These are the numbers listed from previous studies. The most relevant to us was the one in 2021 that had the exact same data set and was using a deep learning and a random forest model. Now, the numbers I'm reporting here are taken over average of multiple runs with random seeds. And what we found was that for a lot of runs, we actually got 100% accuracy on the model where all the 3 classes were accurately predicted by the model. However, once in a while, we would see that the model made an error, and it was always the error for the MCI class, not for the Alzheimer's disease or control class. And because the MCI stage is harder to diagnose, we can understand that it could be hard for the model to predict as well. The next thing we said was, well, does adding this extra attention layers and everything, does it even help? And so we used a no attention baseline. So this was the simple concatenation baseline. And what we found was after hundreds of runs on random seeds, that overall the adding both self-attention and the cross-model attention gave, improved the performance significantly over a simple concatenation baseline. And then the other thing we wanted to see was, well, what modality plays an important role. So after we had trained the model, we just removed one modality at a time in our test set. And one thing that became very clear was that all the 3 modalities were important for the model to make accurate prediction. If you removed any one modality, that tend to create a drop in performance. But the most drastic drop in performance actually came when we removed the clinical modality, suggesting that in the data set that we were looking at with our 200 and so patients, clinical modality actually played a very important role for the model to make accurate predictions in combination with the other 2, imaging and the genetic modality. So that kind of also made us a little suspicious. We wanted to make sure that there wasn't a feature like medicine or something, the medicine that the patient is taking that might, kind of skew this. So we studied the features of the clinical modalities that we were considering. So one thing we made sure was that we removed all the features to our intuition that could be directly connected to the Alzheimer's. But then we also ran a random forest model to get some important scores as to what features were relevant for accurate predictions. And what we found was quite interesting, but not entirely surprising, that the exam scores, like the composite memory score or the language scores, these exams that were done on the patients seemed to indicate or seem to be most relevant for the model when it was making the prediction for the diagnosis. Okay, so that wraps up my second story. So the idea here is that MaDDI is able to integrate 3 different types of modalities to predict the outcome of Alzheimer's disease. We have very explicit interaction modeling in our data set that makes sure that all the interactions between different modalities are considered by the model, and we reduce the cognitive burden by adding attention on top of all the layers. And we report state-of-the-art prediction performance and also reveal that, in the setting that we are considering, clinical data actually plays a very important role. And once again, our code and data sets are available on the GitHub repo if people are interested to use. And so finally, I would like to thank all the students in the lab. It's been 3 years in making, and it's been a wonderful journey. But specifically, thanks to the students who worked on these 2 projects that I talked about. So Jeremy, my PhD student who's now at Agilent, and Mihal, along with the undergraduate students, Shalin and Zavi, and our active collaborators on the project, Erica Larsen, who was involved in the genomics 3D DNA modeling dataset, and then Carsten Eickhoff, who was at Brown University, but unfortunate for us, fortunate for him, has moved on to University of Tübingen now and co-advises Mihal with me. Big thanks to the computational biology core and our funding sources. And thank you for your patience, and I'd be happy to take questions now.
Amazing. Thank you so much, Professor Singh. All right, so as she mentioned, we're going to open the floor up for questions. If you could use the Zoom function to just raise your hand so that you pop to the top of the screen, and I can help moderate. Amazing. All right, Hong Bing, I see you first. Go for it.
Yeah. Hi, very great talk. So I have a question about the interaction between modalities. So for each modality, there's a pairwise interaction with two others. So I think you repeat each pairwise interaction twice. So I think it may cause some redundancies in the final representation. And I wonder, is there any difference between the similar pairwise interaction? Yeah, that's my question.
Yeah, so that's a very good question. And actually, let me try and go back to that slide. Yeah, so we took this, we debated if we wanted to just add one interaction or at a time, or did we want to also flip the order of the interaction that we were trying to see as to what becomes our kind of like query, for example, English language, and what becomes our French language, how do we want to order it? And so we decided to go with that. And that's where we rely heavily on the attention side of things, because the idea is that if one set of interaction is redundant, then the model will weigh that interaction accordingly. So if if it says that I'm getting all the information I need to get from this particular interaction, but not from that, our assumption is that attention would be able to weigh this, because we are allowing the model to weigh only the important interactions, and then the less important interactions, not that much, if that makes sense.
Yeah, got it. Thank you.
Mm-hmm.
Sorry about that, was on mute. Next up, Andre.
Hi, thank you very much for the great talk. My question is about your first project on graph neural networks. And my question is, how were you dealing with the noise in the data? Because I imagine that GNNs are not very robust to given noise.
Yeah, that's a very good question. So we did some, I'm forgetting the exact number, but we did some thresholding on the high-C data, and basically picked, based on that thresholding, picked like very highly frequently interacting nodes, and not, the ones that had low frequency signal, things like that. So we did some pre-processing on the high-C data, which other papers have done as well, to make sure that we only pick the strongest interacting signals and not some of the noise that tends to happen in the high-C data.
Thank you.
Mm-hmm.
Excellent. All right, Gamzee.
Thank you. Well, thank you, Ritambhara. This was wonderful. I have a more general question about the attention-based learning. So my question is about generalizability, right? So when you add attention layer, so I'm not an expert in that, already, but when you add that, it seems to me that you are making your model even more, paying more attention to the data and the details of the data. So I can imagine that, your training data will be captured very well, but when we do these kind of disease predictions, we want our models to work in, one hospital setting to other hospital setting. If it's just learning the details of your training data, how do you generalize these models? And or is it even a problem? Maybe I don't know. So
yeah, no, that's a very good question. I think the idea behind the attention model is to learn the relationship between the features of the data set that you are dealing with, right? So for example, in this, the idea for the cross-model attention is that it learns the importance of how a clinical data interacts with, say, genetic data, and weigh it accordingly. So yes, there is a little bit of, like you could imagine a model learning something very good just based on the training data, but that's where we have all these checks where we do validation and we do testing, and we kept our test set completely held out. The model never saw that, so that we could make the claim that the method is generalizable to the held out test set. Now, the caveat here is that everything was done for the same data set that was processed in the same way. Unfortunately, we don't have access to actual clinical data where we could go and see if it's, a little bit out of distribution, how it would perform. But the attention is more about what propagates through the network, and less about like the reliability on the exact training data. So the regular weights will take care of that, but the attention is more about what relationships are important. And if you expect those same relationships to exist across all your data sets, the model should generalize, but I will not make that claim. It may not. And that's something that we test and we, plan our experiments accordingly.
I guess one could create synthetic data by shifting the distributions a little bit and then try if that data is held out, how it would perform to see if it's generalizable.
Yeah, that's a very good question. I mean, one thing I could do is also see like in language translation, if people have used the same attention mechanism when translating language from one corpus versus the other's corpus. So yeah, some sort of a study like that could definitely help you figure out how generalizable this would be. But it should be very, like technically speaking, but of course, the real world is a whole different story.
Thank you.
Mm-hmm.
Awesome. Alrighty, Jason.
Hi, Dr. Singh. That was a great talk.
Thanks.
For your first project, were the edges of the graph that you showed of the same type?
Yes. So we basically gave binary edges. One means the edge exists and 0 if they were not. Because at that time, my student tried to apply a weighted graph neural networks, and we ran into all sorts of debugging issues. So we kept it simple. But now you do have graph-based neural, and as I mentioned, there are many formulations out there. So now there are graph-based neural network formulations where you can weigh your edges. So you can take this frequency of the interaction and actually assign it as a weight to the edge. And you could do that. And I would assume that might improve the performance.
Okay. God, thanks.
Excellent. Thank you. Alrighty. Are there any other folks with questions? Again, just remember to use the Zoom functionality so I can see you. All right, Shreyas.
Yeah, hi. Thanks for the awesome talk. I had a question about this multimodal project.
Mm-hmm.
I know there's a bunch of these papers recently, particularly in generative modeling of images, like DALL-E and these other papers that use, essentially they encode the two different modalities in a shared embedding space and use some kind of contrastive learning objective.
Mm-hmm.
So I wonder if you thought about using those kinds of methods in this context.
Well, the task in that case would be slightly different, right? Because the task would be you want to predict kind of one modality from the other modality. So for example, the DALL-E, you want to learn the relationship between text and image so that you can generate the image from text and maybe vice versa. This is a slightly different task because the idea here is that we want to learn the relationships between the modalities. So your point is absolutely valid, but the output is not if we can, maybe one day we would want to see if we can generate or we can predict the clinical diagnosis or the actual note from the image and image from the note. We haven't tackled that. Here we are solving a much more simpler task, which is can we just predict 3 categories: control, AD, and MCI conditions.
Got it. Thanks.
Excellent. Alrighty, and are there any other questions? We still have a bit of time, so feel free to ask. Alrighty, I'm not seeing any more hands pop to the top of the screen. So we will take a moment to say thank you so much again to Professor Singh for both a wonderful talk and a wonderful question and discussion session. It was so lovely to have you. Thank you again for your time, and thank you everyone for coming.
Thank you. Thanks for inviting. Appreciate it.